Hello everyone. If you remember, in the previous video we talked about the transcription and translation, and we reached to this slide which is about the purpose of the transcription and translation. Well, of course there are millions of different types of proteins, and the number of the proteins are so high comparing to the genome sequence. And we have different types of proteins and we have different functions. If you look at this picture, you see that there are proteins which are produced for catalysis, muscle contraction, cytoskeleton, blood clotting, and so on. So there is a video regarding to this cute fly and why he produces light. Yes, that's so kind. That's so nice to watch this video together.
How a firefly's tail makes light. First, cells in the firefly's tail make luciferase protein. Then, luciferase carries out a chemical reaction that releases light. The light comes from specialized cells in the firefly's tail. Each cell holds a copy of the firefly's entire genome. One of these genes is a set of instructions for building the protein luciferase. A molecular machine called RNA polymerase makes a messenger RNA copy of the luciferase gene. The gene can be copied many times. The mRNA attaches to a ribosome, the cell's protein-making machinery. The ribosome reads the molecular code of the mRNA to build a luciferase protein, one amino acid at a time. Multiple ribosomes can read each mRNA copy to build many luciferase proteins at a time. When the ribosomes finish reading the mRNA, the proteins are released, and they fold into their final shape. The ribosomes can read the mRNA again to make even more luciferase. Luciferase is an enzyme that catalyzes a chemical reaction between a substance called luciferin and oxygen. This reaction uses energy from ATP, and it releases light. Luciferase can catalyze this reaction again and again. The action of many luciferase enzymes acting at once makes the light bright enough to attract mates and warn off predators.
Okay, did you like the video? So you can pause the video and think about that. As you saw, there was a gene. The name was so cool, luciferase. Did you like the name? Yeah, I love it. It reminds me of Lucifer. I think Lucifer is a very nice name. I don't know in which movie did I see that. Anyway, luciferase is a gene, and the protein which is produced by that gene is responsible for making the catalysis between lucifer proteins which are produced at the end, luciferin, and that catalysis or that chemical reaction produces light. Okay, that was so cool. Let's continue. Yeah, from now on, mostly our review, so we don't have too much talk for the rest of the slides. As you can see, here is the review of the 2.4. Is related to peptide, polypeptides, how they are produced by just a peptide bond, and once the amino acid attaches to the other amino acid, there will be removing of the OH and H from each, OH from one and H from the other one, and production or establishing a peptide bond, and two amino acid makes a dipeptide, which is two amino acids together by the release of the water. So how many amino acids do we have? There are more than 20 actually, but mostly when we talk about the number of the amino acid, this is like it's a old version of the number of the amino acids. So we have 20 amino acids, and some of them are polar amino acids, some of them are non-polar, some of them are positively or negatively charged, and some of them contain sulfur, and we have different types of. So based on these amino acids, also we have different types of proteins. The properties of amino acids determine how a polypeptide folds up into protein. So that's why we have different structures, different shapes of the protein based on these amino acids. So pause the video and think about the properties of proteins. Proteins are commonly described as either being fibrous or globular. We have two different types of the protein in nature. One of them are fibrous and the other one globular. Do you remember what was the fibrous? And do you remember what was the globular? So the globular is, if you want to remember, was something like hemoglobin, which is very famous one as a globular, and fibrous like muscles, elastin, or those kind of things. So yes, pause the video and try to figure out what is the fibrous protein shape, what is the fibrous protein role, solubility, sequence, stability, and examples. Okay? Yes, from now on, pause the video. Okay. Then let's move on. What is the shape of the fibrous protein? The shape of the fibrous protein is long and narrow. The role is structural. They are responsible for strength and support. They are not soluble in the water. Their sequence is repetitive, amino acid sequence. Stability, less sensitive to changes in heat, pH, and etc. So this means that they are more stable than the globular proteins. Example, this is what you have to remember, and this we have already reviewed. This slide is a review slide. Okay, don't worry about that because you have already studied this. Examples: collagen, myosin, fibrin, actin, keratin, elastin. These are all examples for the fibrous protein. Let's move on. Globular. Globular are rounded or spherical. Their role is they have a functional role, including like catalytic or transport, etc. They are more soluble in water. Sequence, they have the irregular amino acid sequence. It's not most repetitive, similar to fibrous. So they have the irregular sequencing. They are more sensitive to changes that make them very sensitive to heat, to pH. And examples are important. Catalase, hemoglobin, insulin, immunoglobulin also are from those globular protein. Let's read this part together. In globular proteins, the hydrophobic R groups are folded into the core of the molecule away from the surrounding water molecules. This makes them soluble. And so, did you get it? Because of the R group, they have the characteristics that they can be soluble in the water because of the charged and the other positivity and negativity. They can be soluble in the water. In fibrous proteins, the hydrophobic R groups are exposed, and therefore the molecule is insoluble. So once this is a hydrophobe, do you remember what was hydrophobe? Means they don't like water. So, and once these R groups are exposed, they don't like water, so they are not soluble in the water. But here, the R groups are folded into the core of the molecule, so the surrounding water molecules, away from surrounding water molecules. So depending on the R group, if they have the folded inside, or if they are exposed to the water, that makes it soluble or insoluble. And also another thing is the nature of the R group also important, because we have different types of the R groups based on their nature. Okay, I believe that this is the last slide, and it's very important, very nice slide. It explains the levels of the protein structure, and each protein is starting from the level 1 or primary structure, and then secondary, thirdly or tertiary, and fourth or quaternary. Pause the video and step by step go and explain, for example, what can you see, what can you explain regarding this structure. Okay, let's go. Primary structure is based on the order of the amino acids, and the covalent bond make that primary structure. What about the secondary structure? If you look at the secondary structure, you see that there are two different types, like alpha helix, which makes a kind of alpha type structure, and then beta sheets. So in secondary structure, the chain of amino acids fold or turn upon themselves. They hold together by hydrogen bonds, and the hydrogen bonds have an important role for making this structure. So there are some examples of the proteins that only have until the secondary structure, like fibrous proteins. What about the tertiary structures? The tertiary structures, the polypeptide folds and coils to form a complex 3D shape. So you see the structure is like a 3D shape, and the interactions are between R groups, including H bonds, including disulfide bonds, bridge, including ionic bonds, and also hydrophilic and hydrophobic interactions. All these types of interactions make that a structure to have a tertiary structure. And tertiary structure may be important for the function because of that specificity of the active site and enzyme. Because the enzyme are all having the tertiary structure, and they have the active site, so there are so many parts of the enzyme which is important for that functionality, and that's why they are much more complex. And globular protein are having all tertiary structure. What about the quaternary structure? If more than one tertiary structure of one protein is attaching to each other, then it makes it for quaternary. So the interaction between multiple polypeptides or prosthetic groups. The prosthetic group is an inorganic compound involved in a protein. That's for example, in hemoglobin, the heme group is like here you can see at the middle of each of them you will find the iron, and that's the meaning of having a prosthetic group. So the interaction between multiple polypeptides or prosthetic groups. Yes. So fibrous protein and globular protein might have the structure for more than one polypeptides together. Okay, that's all for this video. I hope you enjoyed it.